Prior to beginning the activity, please be sure to review the faculty information and disclosure statements, as well as the learning objectives. After listening to the activity, complete the post-test by clicking the Earn Credit link in the episode description. Downloadable slides and resources are also available. The following presentation is copyrighted by Medscape. No use, broadcast, or recording of this presentation or any part thereof is permitted without the written authorization of Medscape.
The following presentation is part of a certified educational activity provided by Medscape Education and supported by an independent educational grant from ViiV Healthcare.
This program is presented by Medscape Education Global.
Welcome. I'm Andrés Finzi, full professor at Université de Montréal and researcher at the Centre de Recherche du CHUM in Montreal, Quebec, Canada. Welcome to this program entitled HIV gp120 Importance Beyond Viral Entry, Potential Role in Immune Activation, Inflammation, and Comorbidities. So in this program, I will cover the role of HIV-1 envelope glycoproteins in viral entry, the role of the gp120 subunit in HIV-induced immune evasion and immune dysfunction, and potential clinical implications. So I like to introduce a little bit how the gp120 is involved in HIV-1 viral entry. So the HIV envelope glycoproteins are a complex that are synthesized in the endoplasmic reticulum as a precursor, which is called the gp160. As it's being transported to the cell surface, it's going to be cleaved into two subunits, the gp120, which is going to end up being the external subunit, and the gp41. The complex will traffic to the cell plasma membrane, and then it will be incorporated into viral particles. The role, so once you have these viral particles, you have around 10 trimers of envelope glycoprotein. So the trimers are 3 copies of heterodimer of gp120 and gp41. So these envelope glycoproteins are required to recognize the target cells. And the target cells for HIV are the cell who has the receptor, which is called CD4. And it uses a co-receptor, which is either CCR5 or CXCR4. The gp120 subunit, which is the external subunit, will engage with CD4. So if you have on the top the viral membrane, so the viral particle will come from the top, and the cell target is in the bottom. When the gp120 engages with CD4, it induces conformational changes, which means that the envelope glycoprotein starts to change its shape. And step by step, it starts to expose different components which are required to engage with the co-receptor, in this case CCR5. At the same time, as you can notice, upon CD4 interaction, you can see that a region of the gp41, the other subunits of this envelope glycoprotein complex, will start to be deployed. And this is very, very important because upon CD4 and co-receptor interaction, there is a stretch of residues here, which is called the fusion peptide, that will be embedded in the target cell membrane. So if we clean a little bit here, this slide, for the first time in the infectious replication cycle, at the first step, what you have is you have a viral component, namely the gp41, which is linked the target cell membrane to the viral membrane. Something that it is important to know is that the gp41 HR1 of the gp41 is complementary to HR2, so these two parts of the gp41 will come together. And by doing that, they're going to be bringing the target and viral membranes together so that the fusion pore can form, and then the virus can transfer the genetic material to the target cell. So this is the role of the amyloglycoprotein of the gp120 in viral entry. But something that is perhaps a bit less known about is that the gp120 can be spontaneously shed and released from the surface of virions and infected cells. Over the years, my group and others have shown that actually the virus makes a lot of effort to make sure that the infected cells is protected from antibodies which are normally present in the plasma of people living with HIV. The virus does that in different ways. But one of them, and it is clear now, is that it's able to keep this trimeric complex of the amyloglycoprotein closed. So the envelope, by being closed, is avoiding a lot of these antibodies that are normally elicited. But in a sense, for the virus, it appears that that may not be enough, because what it does is that the gp120 will be dissociated from the envelope complex, from the gp41, and will be shed. But this shed gp120 keeps its capacity to engage CD4. Please remember that on the amyloglycoprotein complex, the gp120 is responsible to engage CD4. The shed gp120 keeps this capacity of binding CD4. What cell have CD4? Well, CD4 T cell have plenty, and uninfected CD4 T cell have plenty of CD4 at the cell surface. The shed gp120 will engage with CD4, will be stabilizing what we call the CD4 bond conformation, and the gp120 will be exposing epitopes that are highly immunogenic and actually are extremely well recognized by antibodies present in the plasma of people living with HIV. And we and others have shown that when an uninfected bystander cell is coated with shed gp120, these antibodies can engage and can flag this uninfected cell to effector cell from the immune system. So somehow the virus is able to redirect the immune system to kill healthy cells, and at the same time it protects the truly infected cells, which are the ones that are making infectious viral particles. So you do have a redirection of the immune system to target uninfected cells. Something that is important to mention is that there are several antibodies that are able to bind the CD4 bound gp120, which is bound on uninfected bystander cells. But one class of antibodies are very well known, are called anti-cluster A antibodies, and this includes a very well known A32 antibody. So these antibodies are known to be able to kill this uninfected bystander cell. So we know that HIV is a chronic inflammatory condition, even with undetectable viral load under antiretroviral therapy. We also know that the virus is able to persist in diverse reservoirs, in lymphocytes, lymphatic tissues, in the CNS. And we also know that this ongoing chronic inflammation may be linked to an ongoing antigenic stimulation. We know that despite undetectable viral load in the presence of antiretroviral treatment, immunological parameters are not always normalized. ILC levels, D-dimer, the ratio CD4-CD8, several markers of immune activation are not normalized in a subset of people living with HIV, despite success for antiretroviral therapy treatment. And in some of these individuals, comorbidity can happen several years earlier, including cardiovascular, cancer, liver, kidney, and diabetes. So we know that people living with HIV, despite undetectable viral load, a subset of these individuals are suffering from a chronic inflammatory conditions, and this is likely due to persistent antigenic stimulation. And some data that we have been generated in the last few years suggests that the soluble gp120, this gp120 that is being shed from infected cells from viral particles, could be associated, could be part of this problem, may be sustaining this chronic immune dysfunction, and may associated with an increased risk of comorbidity. So the question is, how can we detect soluble gp120 in the plasma for people living with HIV, even in the presence of ART? And I would like to mention this paper, who was published, reported back in 2010 by Jenna Richards, several authors, including James Robinson and Eric Rosenberg. In this article, at the time, so they are following here one individual living with HIV, and you can see the viral load here is shown in white. And upon antiretroviral treatment, viral load becomes undetectable here. And they develop in this paper an assay to measure soluble gp120. I will come back to this assay in one slide. But what is important is that despite viral load control, you can see that the soluble gp120 shown here in black is persistent. So that means that the presence of soluble gp120 is not absolutely linked to the presence of viral load. So this gp120 is coming from somewhere else, and we believe it's coming from some type of reservoirs, and we are highly studying this possibility. But in people living with HIV, where they detected soluble gp120 in plasma, they observed an increased inflammation marker, including TNF-alpha, at the baseline and even during our treatment. So you can see that the gp120, TNF-alpha is higher, statistically significantly higher in gp120 positive individuals versus those who do not have this soluble gp120 detectable in the plasma. So in the lab, what we did is that we adapted the assay reported back in 2010, where we use an antibody which is called C11. C11 is an antibody that kind of grabs the leg of the shed gp120. So we know we are capturing the shed gp120, and we are visualizing the gp120 that is being captured with an antibody that recognizes the CD4 binding site. The goal of the assay is to detect the gp120 who still has the capacity to engage CD4. Okay, and so by doing that, we were able to detect gp120 in around 30% of people living with HIV despite viral load control. And we reported earlier this year at CROI 2024, we did a similar experiment that was reported back in 2010. We follow here two participants. We detect viral load, and then upon ART treatment, viral load will be decreased. And then we can see that the gp120, despite viral load control, which the antiretroviral therapy treatment is in this gray box. You can see that despite controlled viral load, we do detect detectable gp120 over time. And I will come back to that, how this is associated with CD4 cell counts in a few slides. But what I want to mention here is that, again, we see this disconnect between viral loads and the presence of gp120. So the gp120 is coming from somewhere, not necessarily from the viruses that we detect in plasma, but most likely from the reservoir. And so why we are so interested in this gp120 and how this soluble gp120 may be associated with immune activation and dysregulation. And some work that we did in collaboration with Daniel Kaufman a few years ago, something that we found out is that if you make recombinant gp120, the gp120 induces a cytokine burst by monocytes. And we can actually observe that these monocytes, which are being treated with gp120, will produce higher level of IL-10 and IL-1 beta. And we detect this at the cellular level, but also in the secreted cytokine. So gp120 is able to induce monocytes to secrete a lot of inflammatory cytokines. And we reported in this work that for this to happen, the gp120 has to be able to engage CD4. So it appears to be a signaling mechanism through CD4 engagement. We recently follow up on that, where we follow different cytokines, IL-1 beta, TNF alpha, IL-10, IL-6, etc. And here we look at 3 different donors, and each time we add gp120, which here is the supernatant from envelope expressing cell. This supernatant, we were able to quantify the amount of gp120 that was there. And what we observe is that in all the donors, the presence of, when we add soluble gp120, we detect this upregulation of inflammation markers. So if we go back to analysis using plasma from people living with HIV with an undetectable viral load, I insist we detect gp120 in around a third of these individuals. And where we detect gp120, with soluble gp120 in plasma, we also detect increased inflammation markers. In this context here, we are talking about IL-6. So we are pretty much here observing the same phenotype that was reported in 2010. But how this is associated with other potential immune dysfunction, which could be CD4 T cell depletion. So let me go back to what I presented at the beginning. The virus wants to keep the infected cell protected from this immune response, which is called antibody-dependent cellular cytotoxicity. This is when antibodies bind their antigens. So it's kind of a way to flag uninfected cells to the immune system. And then you have effector cells, such as natural killer cells, that will come, detect these antibodies that are binding a viral antigen, for instance, in this context. And then they will kill normally the infected cell. But it looks like the virus is so afraid of this happening that first, the infected cell is resistant to ADCC. So antibody-dependent cellular cytotoxicity stands for ADCC. So the infected cells is resistant to ADCC. We and others have shown that. And among the several mechanisms that the virus uses to protect the infected cell from ADCC, it keeps its trimeric envelope glycoprotein complex close. By being close, the epitopes recognized by these antibodies are hidden. So the antibodies cannot bind, cannot flag out the infected cells. So these cells, which are infected and making productive viral particles, these cells are protected from ADCC. However, the virus is shifting the attention by making soluble gp120. This soluble gp120 still has its capacity to engage with CD4. An infected CD4 T cell have plenty of CD4 out there. The gp120 will recognize CD4, will bind. The gp120 will be stabilized in what we call the CD4 bound conformation. And in this conformation, the gp120 is very well recognized by a family of anti-gp120 antibodies, which are called anti-cluster A antibodies, such as A32. So these antibodies actually end up making a bridge between the uninfected bystander cells and the effector cells, such as NK cells. Again, NK is for natural killer. So these NK cells are taking out this poor uninfected bystander cell while the productively infected cell keep making viral particles. And we can do that in vitro, of course, but what we observe when we look at plasma from people living with HIV with not detectable viral loads is that those individuals who have higher level of soluble gp120 in their plasma, well, we have an inverse association between the presence of these antibodies. The more of you have of these antibodies, the less CD4 T cell counts you have. So it seems, at least we have an association between this soluble gp120 and CD4 T cell count, an inverse association. How this is modulated over time? So I'm gonna go back to these two participants that I presented a few slides ago. In those participants, again, they have viral load. Upon antiretroviral therapy treatment, viral load will be controlled, will become clinically undetectable. And despite this undetectable level of viral load, we still have soluble gp120, which is being modulated. Why is being modulated? We don't fully understand. We believe that this is associated with the size of the reservoir, and we are trying to understand which reservoir. Is this replication competent reservoir? Is the total DNA integrated? So we don't fully understand it as of now, but we're working on that. And so this is the result I just presented one slide ago. We do detect more inflammation marker in people living with HIV with detectable plasmatic soluble gp120. But the other thing that we observe is that in these individuals, we observe a depletion. If we follow up on this longitudinal analysis that we reported earlier this year at CROI, and not only we follow viral load here on the left panels and soluble gp120, plasmatic soluble gp120, as I should show one slide ago, now we can follow the fluctuation of CD4 T cell counts in the middle panel. And I will overlay this for you. If you overlay something that we observe in only two individuals here, right, is that when you can see here in this top panel graph that when the gp120 go down, CD4 T cell count are detectable. When the gp120 goes up, CD4 T cell count go down. So you have this kind of inverse relationship here on participant number 2. You have the same inverse association when the gp120 will go down, CD4 T cell count will go up, and vice versa. This is what we presented earlier this year at CROI, only in 2 individuals. But we have been adding additional individuals to our analysis. As you can see here, we have 16 additional individuals where we observe the same association. Despite viral load control, when we detect plasmatic level of soluble gp120, we observe a decrease in CD4 T cell count. We cannot call this a causality. This is just an inverse association. But it does suggest that if the gp120 is indeed associated with CD4 T cell depletion in vivo, it does suggest that perhaps if we could target the gp120, if we can block the capacity of the gp120 of engaging with CD4, we could potentially restore CD4 T cell counts. And so we believe that soluble gp120, the one that we detect in plasma, act as an effect modifier. So we measure different inflammation markers shown here in green, and these inflammation markers, normally they stick together when we don't observe much association with clinical parameters on the right side of this circle. But as we stratify by the presence of gp120, we start to observe that some associations start to being made, and then some cytokines start to be associated with clinical parameters, including CD4 T cell count. So it's kind of the gp120, stratified by gp120 enables us to put glasses and suddenly start to see some association which are potentially clinically relevant. So it is known that some people living with HIV, despite viral load control, may suffer from atherosclerotic plaques, and they can be detectable in some of these individuals. And we did observe an association between the size of this non-calcified plaque and the presence of soluble gp120, particularly when they are in context of the presence of these anti-cluster A antibodies. So the presence of this viral antigen, which is soluble gp120, and some of the antibodies that recognize it, is associated with the arteriosclerotic plaques that can be detected in some individuals. So, in conclusion, soluble gp120 is detectable in the plasma of around 30% of people living with HIV taking suppressive ART. In those individuals with detectable soluble gp120, we observe that the level of anti-cluster A antibodies, remember, these are the antibodies that are very good at killing these uninfected bystander cells. So the level of these antibodies are associated with a decline on CD4 T cell counts and CD4 CD8 ratios. And we also observe an inverse association between soluble gp120 level and CD4 T cell counts. In these individuals, we observe that several inflammation markers, several pro-inflammatory cytokines, including IL-6, are higher. And also the coronary artery plaque may be larger. So all of this together raise the question whether we could target soluble gp120 to reduce inflammation and curb comorbidity risk. And so there is a clinical trial which is being led by Dr. Madeleine Durand here in Montreal, which is addressing exactly this question. If we use a drug that can specifically target the gp120, can we reduce inflammation? So we know that we can do that in vitro. So we can take the gp120. If we can target gp120, we can block the capacity of gp120 to kill uninfected bystander cells. We can also block the capacity of gp120 to induce a cytokine burst from monocytes. But will this happen in people living with HIV? Well, this RESTART clinical trial, which is being launched in 2025 in Montreal, will address this question. Thank you for participating in this activity. Please continue on to answer the questions that follow and complete the evaluation.
This program was presented by Medscape Education Global.